Hi, my name is Jeff Bennett, and I'm a PhD student in Carol Robinson's lab. And today I'm here with my colleague and supervisor, Corinne. Today we're going to be talking about a recent publication, which should be just out in Nature Chemistry. So we'll start talking about the publication, I guess, Corinne. What was really the driving force behind you wanting to investigate and what did you investigate in this recent study?
Yeah, so the motivation behind the work was that, native mass spectrometry is exceptional at being able to track how proteins talk to other proteins or how they interact with ligands or other drugs. And as we move towards more native environments, things like across the cell membrane, we're interested in how membrane proteins interact with those things. But membrane proteins in mammalian systems or in humans are modified by post-translational modifications, and they start to complicate things because they do influence how those proteins talk to other molecules in the cell. And so even though with native mass spectrometry, we can measure the masses of those complexes with very high accuracy, we don't always know what those complexes are made of.
How did you actually work out what's within these really complex environments?
So we had to develop a tool called native top-down mass spectrometry, in which the native complex is selectively isolated, and then we can break that complex into substituent parts, and we can then learn about those segments of the parts of the complex.
I'm just really interested to know what were the main obstacles you faced and how did you overcome them?
Yes, so the major obstacles first was, because we're interested in membrane proteins, was getting them into the mass spectrometer. As the membrane proteins have this additional challenge where they're hydrophobic and they need to be stabilized by the membrane, but that hinders our mass analysis. And so we needed to develop a means to strip away the membrane bilayer and keep the protein. Typically, in those sorts of experiments, to do this in the mass spectrometer, you utilize a method called collisional activation, in which you essentially throw this protein complex into a wall of gas to shake off the lipids. Up until this point, we were utilizing or maxing out all of the available collisional activation that we could get to just get the protein out of the membrane, to be able to analyze them. And so we needed to come up with a new way to both get the membrane protein out of the membrane, but then also have enough activation left over to break it. And so one of the things that we did was team up with Thermo Fisher Scientific in San Jose, California, where we modified an instrument to incorporate an infrared laser. And so what this laser does now is we can tune the intensity and the time at which we're irradiating the membrane. So we can gently remove the membrane bilayer and release the protein, but then we're left with the ability to then, hit the protein again with even more laser power, and then break it into those subsequent parts, which we can then analyze the composition. This was a really good collaboration here at Kavli as well. We worked with Weston Struve's group and Dr. Sean Burnap, and it is one of the challenges is that glycans and proteins, fragment in a very specific way. And so having their glycoproteomics expertise on this was invaluable.
What were the main findings and how will that affect everyone all around us?
So one of the things that we used in this study was we took raw disc membranes from retina, as these membranes are some of the most well-characterized membranes out there. We know how visual phototransduction occurs. But what we're really surprised to find is that we found new forms of the proteins that had never been discovered or annotated before. And we found a lot of really unusual post-translational modifications that ended up mattering as in terms of influencing how those proteins came together and talked to one another. And so one of the cool things that we were able to do was we were able to challenge these membranes then with drugs. We used phosphodiesterase inhibitors that are originally targeting a phosphodiesterase in blood vessels elsewhere. But these drugs typically have a lot of side effects involving vision. And so we hypothesized that these drugs could be interacting with a lot of the proteins that are in the retina. And to our surprise, we did find a lot of off-target drug binding of these drugs to retinal phosphodiesterase 6. But we also found off-target drug binding to G proteins and other signal transduction proteins. But it wasn't just off-target binding. These drugs actually preferred to bind with lipid-modified forms. And so now, for the first time, we're actually seeing a drug interaction specific to a post-translational modification or a certain form of a protein. And so this was really exciting for drug discovery efforts and trying to mitigate adverse side effects.
what other sorts of molecules or modifications did you discover within your rod system?
So we discovered a lot of lipidation, and these lipid modifications are actually really important because they help localize these proteins to the membrane where they need to enact, their action signal with other proteins. The interesting finding is that we didn't just find, one flavor of lipidation. We found a whole range of different lipid types, and different levels of unsaturation, things that were very specific to the proteins that we were analyzing.
Okay, and so now you've established this platform and shown it can work, I guess, in a system that's been studied, but yet you can discover so much more about. What are you hoping to apply it towards, and where do you see the research moving?
I think the most exciting bit about this is that this can be applied broadly to just understanding how post-translational modifications on a protein simply dictate those interactions and what's driving how proteins signal or how they function in our bodies. But the other exciting component is that we can now apply this to drug discovery efforts. And so we can better understand how drugs specifically bind to certain forms of proteins. And we can also learn about off-target effects, so unintended interactions with certain proteins and sort of the drivers that would influence that.